Hello and welcome to the video tutorial of SIQPlotteR. This is the module that allows you to visualize the analysis done by SIQ on your amplicon targeted sequencing data. So SIQPlotteR is basically an R Shiny application, and because of that it's interactive, and we designed it such that here on the left side you have many different options, which I will just go through quickly as soon as I have loaded the data. Normally what you do, you browse to the Excel file that has been output by SIQ, or you drag and drop it here. Alternatively, there's an option to load all the data which has been analyzed for the SIQ paper, and it will show you directly all the tornado plots. We'll come to that in a short moment. First, here on the left you have the options to select which mutations you want to actually see. Normally all are visible. Then you can select the samples that you want to actually visualize, and that is also connected to the target. In this case there are 6 different targets being simultaneously visible, and I'm going to change that slightly because I only want to first look at Cas9 wild type events and only the ones in figure 2, to give you an indication of how these mutation profiles look. So one of the plots we made is called a tornado plot, which is visualized over here, and what you see in colors are the different classes of mutations combined with the microhomology. So if you have, for example, a deletion with a microhomology such as this one, it will actually show you that it has 6 base pairs of homology, and the majority of events is actually that in the Q80 deficient background. As you can see, the samples are sorted alphabetically, which is not always handy, so we have also the option in SIQ, and that's completely down to actually sort them. So normally you would like to see the wild type first. You just drag it to the first location, and now you see that wild type is actually the first sample to be shown. If you, for example, find that the size is too small or too big, you can of course always change that. And what is also, I think, is neat is that you can actually alter this plot by sub-selecting certain types. So let's say you want to only look at deletions, then you can see that wild type cells here have a very specific pattern which is almost identical in the Q80 background. But if you then look at pol theta deficient sample, or cells which are pol theta deficient samples, we see completely different deletion spectrum. What you can also do is change the number of columns. In some cases that's very handy, and if you want to zoom in a little bit on where all the mutations are, you can of course also do that. And what I haven't explained yet is that the white part here represents the deleted part, and then the edges are the flanks which are still present in the read. You can change that by changing the sort options and changing the tornado type. And one thing I will show you first now is that what if we want to look only at deletions? Then the tornado plot becomes 1 for all the samples, but of course we're looking at a subset of all the reads which has been analyzed by SIQ. So what if we then instead use, in this case, the y value not set, that we see that in wild type you have about 80% of reads present, and in Q80 that's maybe slightly less. And then there's a very nice option called max of plots, which uses the maximum of all the samples included in the plot and sets the y-axis accordingly. So here the highest is Q80, and then the wild type in the pol Q has been set relatively to that sample. You can sort the tornado plot also on the position, the center of the event, the size of the event, which in some cases makes more sense. Type doesn't really do much here because we only select the deletions. I can of course change that again. So here you can simply play around with the sorting options. Size is also a nice option, I would say. And then you can also change the tornado type. So this is a regular tornado type, but you can also invert it, and then you only see the deleted part of the plot. In some cases that's very handy, in other cases maybe less so. There are some other options here which are experimental and might disappear in follow-up versions of SIQPlotteR. You can also show the number of reads, which in some cases is handy, just to get an idea of how many reads are there. However, this information is also present here in the title. So here we see about 8 million reads, which is way more than you actually need to get a proper mutation profile. This is just visual representation. If you don't want to have this this white part in the plot over here, then you can actually remove that. And finally, you can export this graph. And if you then click it, you can actually do something with these plots. For example, use them in Illustrator. I spent quite a bit of time already on the tornado plot, but there are different types of plots for all the available within SIQPlotteR. So allele plots is a novel plot which shows you the top X outcomes. In this case, the top 10 outcomes which are present in these 3 samples. And as you can see, as in the tornado plots, that the top outcome is a 14 base pair deletion with a 6 base pair homology in there. And that's mainly present in the wild type and the Q80, and not so much in the pol Q cells. You here see the top 10, as I said. You can change that by very simply looking at, for example, the top 5 or even the top 100 if you would like to do so. Underneath you also have the table, which then alters. The first thing shown is always the reference, and this mark over here is the location of the expected cut site, which you have set in SIQ using the left and the right flank. And then here this symbol over here, the accolade symbol, that represents the insertion location, and in this case the A is inserted at this location. So another way to visualize that is just looking at all the types found. As you can see here, the majority of events are deletions in these samples, and here you don't see overt differences between the samples. Therefore, you really sometimes have to look at the tornado plots, but sometimes the type plot is already enough to give you the information that you require. Also here, what is shown is a table with all the frequencies, which you can change. Always check here on the left side which options you have, because they change if you have a different plot open. So microhomology, that's something that we in the lab look at quite regularly. In this case, we see that wild type and Q80 are very comparable. Much microhomology is being used, and for the microhomology only the deletions and tandem duplications are included for the plot. And here you see in pol Q sample that clearly there's less microhomology being used. So here you can look at the sizes. In this case, the deletions are plotted in the minus direction, but that can be also changed by using here. If you want to only look at the deletion size, then suddenly they're plotted in the plus direction, and now it's plotted as a heat map representation. So here you see the large deletion with the microhomology, which is not so present in the pol theta background, and here's another deletion with some microhomology, which is not, again, almost not present in the pol theta background. You can also use violent plots, but then you lose in some cases some of the details that you get with the heat map plot, and vice versa. We also have a representation of having SNPs. In this case, it doesn't give us a lot because there's no, there are almost no SNPs being found. So I changed that to a base editor sample that we have included in the plot or in the paper, and here you see that suddenly we do see lots of SNPs at this location. One filter I haven't explained yet to you is that in some cases you only want to include events that overlap with the expected cut size, and this is what this filter actually does. You can set it to 10 base pairs or 2 base pairs. In this case I will set it to 10 base pairs. I think, yes, the SIQPlotteR will reload, which unfortunately sometimes happens. and now you see only the SNPs from -10 to 10 base pairs. So you see there's a predominant base change at this position from 0 to a T. If you really want to see what it is, you can of course switch to the allele table. And what we've also noticed is that sometimes the SNPs are actually dinucleotide changes. So with this slider you can actually set the SNP size to 2. So that means that we're also including SNPs which have altered 2 base pairs, in this case change it to TT. If we go quickly back to the allele plot, you will see that there's indeed the deletion of this CC in the reference, and that has become TT, and that is the most predominant outcome here. Even more so than the 1 base pair SNP, which is in second place. So here you can see the target alteration plot, so which base is mostly targeted in which sample. That can provide you with some insight as well. Sample info is actually very handy to discuss, so I will do that briefly, and I'll switch back to the Cas9 wild type samples in this case. What you get here is a plot of the fraction of reads in each sample which are correct, which is represented by the black bar. The gray are the reads which couldn't be merged, and then the red ones are the ones that failed various filters, or at least one of them, and are thus excluded from the final analysis. If you see any problems here, it might indicate, if you see very low correct reads, for example, it might indicate that either your initial settings for the primers or the flanks are not correct, or for example there's a problem with the reference sequence that you have given as input to SIQ. So always check these sample information plots, because that will give you some information about whether or not your analysis went well. So this is a relative plot, but you can always do an actual absolute plot, so where you see the actual number of reads, and then in this case you see that for a wild type sample we have about how many is it? 5 million reads which are correct, which is way too much to get a correct mutation profile. You can do with a lot less reads. The other plots I want to discuss not at the moment. However, if you want to check some additional information, also linked to the user guide, you can always go to the About tab and find more information there. Thank you for joining us on this tutorial.